Hello, everyone. Today we're diving into a crash course on the cellular basis of reproduction and inheritance. We're going right to the heart of biology, really, to the most fundamental action, cell division. It sounds so simple, but it's really life's great paradox.
It is. It's this mechanism that's so crucial.
Yeah.
Without it, you wouldn't exist. But at the same time, it holds this potential for catastrophic failure.
Right. It's a double-edged sword.
Absolutely.
Yeah.
I mean, think about what it does for you right now.
Yeah.
It takes one single cell, a fertilized egg, and builds a whole person.
Mm.
And then it's your body's maintenance crew.
For renewal, repair, all of that.
Yeah, constantly. Your body's replacing millions of cells every single second.
Mm.
Just think about your skin cells alone. That requires constant, and I mean flawless, division.
And that's the edge of the sword, right?
Yeah.
If that process, that essential process, loses its control even a little bit, the results can be just devastating. I mean, uncontrolled division isn't just a mistake. It leads directly to diseases. Cancer is, of course, the big one.
It is. The whole system has to be so precisely regulated. And it all starts with two fundamental pathways the cell can take.
Okay.
So first, you have what we call asexual reproduction. This is the path of, well, sameness. A single parent creates offspring that are perfect genetic clones.
And you see this everywhere. A sea star regrowing an arm.
Right.
Or yeast budding. Or even just taking a cutting from a houseplant, like an African violet, and a whole new identical plant grows. That's a clone.
Exactly. But then there's the other path, the engine of evolution, sexual reproduction. This one is all about complexity.
It needs two specialized cells, the gametes, to fuse.
Right, the sperm and the egg. And this path is built for variation. The offspring get this unique blend of genes from two parents, so they're not clones, they're variations on a theme. And to understand how a cell can do both of these things, we have to start with the cell cycle itself.
Okay, let's unpack this. So the life of a cell isn't all division. Most of its time is actually spent just preparing.
That's it, exactly. The vast majority of a cell's life, maybe 90% or more, is spent in the stage we call interphase. And this is a time of really intense activity. The cell is growing, it's doing its job, and it's replicating.
But interphase isn't just one long waiting period. It's structured, right? We break it down.
We do. It starts with G1, or the first gap. Yeah. This is where the cell just grows and does its normal functions. But G1 it's the decision phase. There's a critical checkpoint at the end of it.
And this is where it gets really interesting. That checkpoint is basically the cell asking itself, am I ready? Do I have the resources? Did I get the go signal to divide? And if the answer is no, some cells, like our mature nerve or muscle cells, they just exit the cycle. They go into a non-dividing state called G0.
Right. That's the point of commitment. If it gets the green light, it enters the S phase. S stands for synthesis.
Synthesis of DNA. This is when replication happens.
The only time replication happens. And it's so important. Your single chromosomes become duplicated chromosomes.
And after that, there's one last prep step, G2, the second gap.
Yeah, the cell just finishes making all the proteins it needs, making sure everything is absolutely ready for the main event.
Which is the mitotic phase, or M phase, the last 10%.
And that has two parts.
Sure.
First, mitosis, which is the division of the nucleus, and then cytokinesis, the division of the actual cytoplasm. And, this process is just amazingly accurate. In yeast, for example, they see an error maybe once in every 100,000 divisions.
That's incredible precision. And it's how every cell in your body has the same 46 chromosomes. But to move all that genetic material around, you need some serious organization.
Absolutely. I mean, normally in interphase, the DNA is just this long, thin, diffuse stuff we call chromatin. It's way too thin to see or move around efficiently.
It's like trying to move miles of thread around a room without tangling it. It would be a total mess.
It would be a disaster.
Yeah.
So before it divides, the cell does this incredible packaging job. The chromatin fibers coil and fold up until these thick, distinct structures we can actually see. We call them chromosomes.
And because this happens after the S phase, what we're seeing is a duplicated chromosome. It's made of two identical copies, right? The sister chromatids.
Correct. And they're held together most tightly at a little pinched-in waist called the centromere.
So that duplication is the key to sameness. A human cell starts with 46 duplicated chromosomes.
And when those sister chromatids split, each of the two new daughter cells gets a perfect, complete set of 46 single chromosomes. It's a really elegant solution.
So now we get to the actual dance of mitosis. We break it down into phases, and it's all driven by this machine called the mitotic spindle.
Which is basically the cell's internal cable system. It's made of microtubules, and they grow out from these structures called centrosomes.
Okay, so first up is prophase. This is where we start to see the action.
Right. The chromatin finishes condensing into those visible chromosomes, and you see the spindles starting to form as the centrosomes move to opposite ends of the cell.
Then comes prometaphase.
And this is when the nuclear envelope, the membrane around the nucleus, just breaks down. It dissolves. And that opens up the arena for the spindle microtubules to attach to the chromosomes.
They attach to a specific protein handle, right, at the centromere.
They do. It's called a kinetochore. And that connection is absolutely critical. Think of it like a docking station. If the cable misses its dock, that chromosome is going nowhere.
So if the attachment is good, we move to metaphase. This is the one everyone recognizes from pictures.
It is. All the duplicated chromosomes line up perfectly single file right along the cell's equator. We call it the metaphase plate.
And then the moment of truth, anaphase.
This is it. The centromeres that hold the sister chromatids together, they split. And instantly, those chromatids are considered their own individual chromosomes. Motor proteins then start pulling them to opposite ends of the cell.
And the whole cell actually stretches out, it elongates.
It does. And mitosis wraps up with telophase. This is basically cleanup. New nuclei form at both poles, the chromosomes uncoil back into chromatin, and the spindle disappears. The nuclear division is done.
But the cell isn't two cells yet. We still need to divide the cytoplasm. And that process, cytokinesis, is totally different in an animal cell versus a plant cell.
Completely different. In animal cells, it's done by cleavage. Because they're soft, a groove forms on the surface, the cleavage furrow.
Like pulling a drawstring.
Exactly like a drawstring. It's a ring of proteins that just pinches the cell in two.
But plant cells have that rigid cell wall. They can't pinch.
They can't, so they have to build a new wall from the inside out. Vesicles full of cell wall material line up in the middle and start to fuse. They form this thing called a cell plate.
And that plate just grows outwards until it connects with the existing cell wall.
Yep. And then it solidifies, and suddenly you have two separate daughter cells, a brand new wall between them.
It's an amazing bit of engineering. But, as we said at the start, this whole thing depends on control. When that control is lost, you get cancer.
Right. Normal cells are disciplined. They listen to signals. Two big ones are anchorage dependence. They have to be stuck to a surface to divide, and density-dependent inhibition.
Which is like a crowding sensor. They stop dividing when they touch each other.
Exactly. Cancer cells are, well, they're cellular anarchists. They ignore all the rules. They don't need to be anchored. And most importantly, they ignore that crowding signal. They just keep dividing and piling up, forming a tumor.
They've lost the ability to listen to that G1 checkpoint.
They've completely lost it. And you get two kinds of tumors. A benign tumor stays in one place. You remove it, problem is usually solved. The real danger is a malignant tumor.
Because those cells can invade other tissues. They can travel.
And that's the key. That process of spreading, of malignant cells getting into the blood or lymph system and starting new tumors somewhere else, that's called metastasis. That's when we clinically call it cancer.
And this is why so many treatments, like chemotherapy, target these very mechanics we've been talking about.
They do. A drug like vinblastine stops the spindle from even forming. But then a drug like Taxol does the opposite. It freezes the spindle after it forms, trapping the cells in metaphase. You're weaponizing the cell's own machinery against it.
That's a really powerful way to think about it. And it's a perfect pivot point. We've talked about how the body makes perfect copies. Mitosis is for sameness. But how does biology create variety? Now we have to talk about meiosis.
Meiosis. This is the engine of sexual reproduction. Its job is to make cells that are genetically unique and, crucially, to cut the number of chromosomes in half.
So we need to talk about chromosome counts. We say our body cells or somatic cells are diploid or two nullars, two complete sets, 46 in humans.
Right. And then our gamete, sperm, and egg are haploid, or 9-nullars. They have only one set, 23 chromosomes in humans.
And that diploid number means we have homologous chromosomes.
The matching pairs. One you inherited from your mother, one from your father. They carry genes for the same traits, like eye color, but they might carry different versions of those genes, one for blue eyes, one for brown.
And meiosis is the process that makes sure the gametes get only one from each pair. It's one round of duplication, but then 2 rounds of division. Meiosis the first and meiosis the second.
And meiosis is the really unique one. We call it the reduction division. This is where the chromosome number gets cut in half. And the most important event happens in prophase A.
This is when the homologous pairs find each other.
They do more than find each other. They pair up gene by gene, physically connecting to form a structure called a tetrad. And while they're paired up like that, they do something amazing called crossing over.
They swap pieces.
They literally swap pieces. The non-sister chromatids, one from mom, one from dad, exchange corresponding segments. They cut, swap, and paste their DNA.
And that right there is the first huge source of genetic shuffling. You don't just get a mom chromosome or a dad chromosome anymore. You get a hybrid.
A brand new recombinant chromosome. It's immediate and it's profound. Then, in anaphase I, we see the other key difference from mitosis. The homologous pairs separate.
Wait, the pair is separate, but the sister chromatids, they stay attached.
They stay attached. That's the key. This is the opposite of mitosis.
And that specific step is what makes the cells haploid.
That's the moment. Because you've separated the sets, each pole of the cell now only has one chromosome from each homologous pair. The set number has been reduced from diploid to haploid.
So meiosis the second is just cleanup then. It's separating those sister chromatids that are still attached.
It's basically mitosis, but for haploid cells. And at the end, you have 4 daughter cells, and every single one is a unique haploid gamete.
The amount of variation the system can generate is just astronomical. It's not an accident. It's designed for it.
Absolutely. There are 3 main sources. First is that crossing over we just talked about, creating those recombinant chromosomes.
Second is what we call independent orientation. This is just pure chance. The way those homologous pairs line up at the metaphase plate in meiosis the first is totally random.
And with 23 pairs of chromosomes in humans, that randomness alone creates 2 possible combinations.
Which is, what, about 8 million?
Roughly 8 million possible combinations for a single sperm or a single egg, just from how they happen to line up.
And then the third factor is random fertilization.
You take one of those 8 million possible sperm,
Yeah.
and it fuses with one of 8 million possible eggs. You multiply the possibilities, you get a zygote with about 64 trillion possible combinations.
64 trillion. That number is just, it's why every single one of us, unless you have an identical twin, is completely genetically unique.
But for a process this complex, errors can happen. It's rare, but they do. The failure of chromosomes to separate properly is called non-disjunction.
And that can happen in meiosis the first if the homologous pairs don't separate, or in meiosis the second if the sister chromatids don't.
Either way, you end up with abnormal gametes, some with an extra chromosome, a nuller plus bolars, and some missing one, an enlunar.
And if one of those fuses with a normal gamete, you get a zygote with an abnormal number of chromosomes. The most common result of this is trisomy 21.
Or Down syndrome. An individual inherits 3 copies of chromosome 21 instead of 2. So 47 chromosomes in total. And we know that the incidence of this increases quite sharply with maternal age.
It's also worth pointing out that errors with the sex chromosomes, like XXY or XO, tend to be less severe than with the other autosomes.
Yeah, that's largely because the body can compensate. It can silence extra X chromosomes, and the Y chromosome carries relatively few genes.
So to really bring this all together, the comparison between these two processes really comes down to their purpose.
It does. Mitosis is for maintenance. 1 division, 2 identical diploid cells. Meiosis is for reproduction. 2 divisions, 4 genetically unique haploid cells.
Okay, let's unpack this. So what does this all mean? We started by saying cell division is this double-edged sword, this incredible balance, and it's so fragile. Most of the time, an error in meiosis is lethal. It results in a spontaneous abortion. But sometimes failure is actually a mechanism for change.
It's true. Occasionally, massive failures leading to polyploidy, multiple whole sets of chromosomes, have actually been a driving force in the evolution of new species. You see it a lot in plants, and even in some animals like the great tree frog.
So failure can, in a strange way, be a feature, not just a bug.
It can be. But at the end of the day, you should just reflect on the immense precision required to keep your own body healthy. Remember, we mentioned those mature nerve and muscle cells. They exit the cell cycle permanently. They enter G0. And that is why damage to those tissues is so often irreversible. Your health relies on the perfection of that cycle, because once that perfection is lost, for some cells, there really is no going back.